Students are your students, I think. That's why they are not asking.
Manos is, yes, Manos. What?
So basically my question is that so many drugs are available in the antimicrobial, and you have shown that presented that different drugs, though they are depolymerizer, but their activity is different. Now that, how do you think that the C12 we can proceed further?
See, this compound is actually good because it is, limiting a lot of antibiotics. But we needed to look at this good toxicology profile and then try to see any, one is you can sell it to any company. And do more human model study and look at the toxicology pattern, and that's the aim of academic lab. We can't go beyond this.
And then my next, though tubulin targeted drug is so much the commercial value, but why the tubulin targeted drug is not the first choice? It is always given in the combination therapy.
No, there are several tubulin targeted drugs are in first choice, and combination therapy, people use combination therapy because that minimize the toxicity. Okay, and if you look at this today, most of the drugs are used in combination. Paclitaxel also used in combination, because of the toxicity only. It's a huge toxicity that is associated with it. Just remember, no kinase, people drum about kinase, but no kinase inhibitor is ever a drug. Okay, even polokinase, no kinase inhibitor is a drug because of, again, the problem is toxicity.
Okay, thank you very much. Last question from my colleague, Dr. Shankar.
Excellent talk. You told that acetylation in biological processes stabilize the microtubule.
Yeah.
So if the design compound has acetyl motif, it can be N-acetyl, O-acetyl, then acyl transfer can happen and then stabilization of microtubule do. So that time acetyl analog will be better. What's your opinion?
People have burned their hand looking at that idea. Okay. A lot of people look at HDAC or acetyl transferase and all these things. Though that all generates a lot of hype. Till today, none of these acetyl transferase inhibitor or HDAC inhibitor actually reached to a stage that is called successful. Because of the, if you look at HDACs, okay, there is at least I know of 6 or 7 different HDAC 6 actually deacetylates tube. Acetyl transferase is now known which acetylate tube. Only deacetylate tubulin is the HDAC 6. But if you look at HDAC 6 and other HDAC, then it is very, very similar. So you don't get a specific inhibitor. And even in test tube, if you get a specific inhibitor, just many of you actually will not complete this thing. I will tell you one thing. If you look at this taxol, you add taxol in cell culture media, 5 nanomolar, cells die. But if you look at intercellular concentration of taxol, it is actually 500 nanomolar or 1000 nanomolar. So almost 200 to 500 times more. So and what happened to curcumin? What curcumin is so far felt. Curcumin will add 17 micromolar, it will kill the cell. Inside the cells, the intercellular concentration will be 35 micromolar, just 2 times or 3 times more. So if the uptake is less, then it will not do anything to the cells. That's why curcumin does not work most of the time. So if you look at the acetylase inhibitor or others, they may have some difference in test tube or cell culture level. But as soon as you give in organism level, say in animal, mice or other, what will happen? because the concentration will increase that level, okay, and then you will not see the difference. So minor difference, so that's why the kinase inhibitors are not working, or many other inhibitors are not working. It is wonder that how do the microtubule inhibitors work, but it does work. So
Thank you, Dr. Dulal, for your wonderful talk. On behalf of organizers, a small token of appreciation. The next speaker is Dr. Bushra Ateeq. Very happy to introduce this wonderful scientist. She is Associate Professor and Joy Gill Chair at IIT Kanpur from Department of Biological Sciences and Bioengineering. As far as awards are concerned, she is elected fellow in the year 2021 of the Indian Academy of Sciences, Bangalore, elected fellow in 2021 of the National Academy of Sciences, India, NASI, and 2020 Shanti Swarup Bhatnagar Award and CNR Rao Faculty Award for the Excellence in Research at IIT Kanpur. With this brief introduction, I request our speaker Ateeq to deliver her talk. Dr. Bushra.
Am I audible? Yeah. So, good afternoon, everyone, and thank you so much, Professor Sharma, for inviting me here and giving me this opportunity to present my work at Naipar Mohali. And thanks, Professor Murthy, for kind introduction. And this is my first trip to Naipar Mohali, and I'm really happy to be here. I have high respect for this institute because one of my students currently who is submitted PhD, she's from here, Anjali. So glad to be here. Yeah, thank you. So, as you saw the title that I'm going to talk about prostate cancer, and this disease mainly affect the men who are at the older side, the average age to get the disease is around above 50 years. And due to increase in age expectancy in India, there is a steady rise in the cases. So if you look at the cancer history data, when in 1990s it was the 8th more common prevalent cancer. But if you look at the data in 2015 and 16, it's marked as the 3rd most common cancer, most prevalent cancer. So this is the schema showing prostate gland, and so this is a schema of showing prostate gland. There are 3 different zones, and all the 3 zones, they have different propensity to develop the cancer. So most of the cancer they develop into the peripheral zone, followed by the transition zone. And this difference is probably that many school of thought, they think it could be probably because of the androgen signaling. Histologically, if you look at the gland, it's one foci, and there are multiple thousands and thousands of such foci in the gland, and there are 3 different kind of glands: luminal glands, basal cells which are lying down the basal lamina, and few neuroendocrine cells. And with neoplastic transformation, most of these luminal cells, they start dividing uncontrollably, giving rise to adenocarcinoma. So as the first, Professor Panda mentioned about docetaxel and other Taxol used in prostate cancer, but in majority of the cases, the first line of therapy is androgen deprivation therapy, where they use either surgical castration or chemical castration. And the reason they use androgen deprivation therapy is because this disease is very much dependent on androgen signaling. The gland development is also dependent on androgen signaling, as well as the disease is dependent on androgen signaling. But the bad news is, after few years, like 4 to 5 years, these cases they progress into castration-resistant prostate cancer. And at this stage, majority of the cases they have this very aggressive disease which no longer respond to anti-androgen therapy. And if you look at multiple aberration into the androgen receptor are present at this stage, like AR amplification, mutation, and splice variant. Not only that, these patients further progress into neuroendocrine prostate cancer, which could be either focal, few focal lesion you can see, or it could be overt neuroendocrine prostate cancer. So if you look at the molecular alteration, like there are multiple and stage wise, when the disease is organ confined, you see gene fusion and mutation in some of the tumor suppressor gene, and like there are lot of genetic rearrangements. And as I mentioned, AR specific alteration and neuroendocrine shows more of different more epigenetic dysregulation, epigenetic regulator, and some of the transdifferentiation marking coming up. So again, the disease is very heterogeneous, and because of this heterogeneity, the clinical outcome of the patient is very varied. Some patients respond nicely to certain drugs, some patients show certain rational mechanism. And one of the most recurrent subtype is TMPRSS2 fusion, and here you see that TMPRSS2, which is an androgen driven gene, contributes the enhancer region to this ERK and structure factor family, and this is about 50% of the cases they have this genetic rearrangement, followed by RAF kinase, which is present in 2% of the cases where you have like CRAF and BRAF, and SPINK1, which is serine protease inhibitory calcium type 1, which is highly expressed in 10 to 15% of the cases, and these patients they show very aggressive disease and shorter biochemical difference. Now, when I started my group, one question was like, what is this molecular heterogeneity in Indian prostate cancer patient, and there was not much information available. So, with the help of my clinical collaborator, we gathered some 121 samples, and instead of going for like expensive technologies such as WS and RNA-seq and all that, we just focus which is like more relevant in clinic, such as like immunohistochemistry for ERK, oncoprotein, which is which as a result of fusion is highly expressed, and validation at the genomic level whether there is a person having genetic rearrangement or not. So this is a case of a prostatectomy specimen, and you can see ERK is highly expressed. Not only that, if you look at this fish data, these two distinct dots, red and green, is showing genetic alteration involving ERK locus. Not only that, about 10 to 15% of the cases they show SPINK overexpression. So this is the data we got from Indian cohort. So one thing that caught our attention was RAF kinase, which was only 2% in Caucasian cohort, was around 6% for Indian prostate cancer patient. Now, knowing that like in this disease most of the time like there is needle core biopsy, like at least in India, like there is no such non-invasive diagnostic test available, but in many European and US country, like US, you can see like these kind of urine based biopsy tests. So here what they do, like they either use urine sediment or like they isolate exosome, and they look for certain marker. The marker that I told you, TMPRSS2 ERK and PCA3 is a non-encoding RNA, DLX1 and HOXY6. So one thing interesting about DLX1 is like it's a gene which is involved in development. It's involved in differentiation of the GABAergic neuron of the basal ganglia and cerebral cortex. Not only that, it's also involved in the craniofacial patterning and the mandible jaw development. And it's true for many cancer genes. They are known to be playing very critical role in embryonic development, but later on in cancer, they again abruptly overexpress and then they drive the cancer. So with this, one of my students, she asked like the question like how DLX1, if we could detect into the urine, in the urine sediment, what is the expression of DLX1 in localized and metastatic prostate cancer? What is the mechanism? Why embryonic stage is there, but why in the prostate cancer is coming up? And if it is coming up, what functional role it's playing in disease progression? So with this, what we did, we collaborated with Dr. Nalla, and what we did, we stained around 124 prostate specimen, which were like more of a naive, they were not treated with any drug, and we looked for the expression of DLX1, and we found around 60% of the prostate cancer patient, they show DLX1 expression. This is an in situ RNA like staining, and these red dots represent the RNA transcript for DLX1. Not only that, since I told you like majority of the 50% of the cases they overexpress, they show genetic alteration between TMPRSS2 ERK, so ERK is high, then the obvious question is like if it's 60% present, what is the connection between ERK and DLX1? And what we found that 96% of the ER positive patient, they also have DLX1 overexpression, and about 50% of the cases or 42% of the cases, they are though positive for DLX1, but they are negative for overexpression. So there is a positive correlation for DLX1 and ER. Moving forward, since I told you that androgen signaling play critical role, so the obvious question is like what is the connection between androgen receptor signaling or androgen receptor in DLX1 positive cases? Sorry. So when we looked into that data, we again stained for the same set of patients for androgen receptor immunohistochemistry and ER, and what we found that majority of the ER positive patients, they show AR staining, they are positive for androgen signaling. Not only that, like around 50% of the DLX1 positive patients, they show overexpression of ER. When we look into the clinical data, majority of the patients which have higher Gleason score, which is a pathological grading and considered to be much more aggressive if the Gleason score is high, they positively correlate or they show much more higher expression of DLX1 at advanced stage of the disease. Not only that, when we look at the different staging, again DLX1 at high and the medium level show much more association with the pathological stage of PT3A and P3B. Sorry. Now, since I already introduced you to castration-resistant prostate cancer, the next obvious question is how the disease look like or how like in CRPC stage DLX1 expression is. And by collaborating with COM or S.A.S at University of Washington, we got this metastatic biopsies from bone, liver and lung and lymph node and others, and we found that majority of the patients which have bone metastasis, they tend to show higher DLX1 expression followed by other organs such as liver, lung and lymph node, which again indicate that DLX1 overexpression correlates with much more aggressive disease. And not only that, the patient who develop bone metastasis, they tend to show much more higher expression of DLX1. Now, going back to the laboratory experiment, so the second thing is like how it could prove that DLX1 indeed play critical role in tumorigenesis. So what we did, we used this RWP cells, which are non-cancer cell line. These are immortalized epithelial cells, and we overexpress DLX1, and with overexpression we can see there is a change in oncogenic potential. These cells show increase in proliferation, more foci formation, more cell migration or invasion. Not only that, when we use another approach, loss of function assays, in another cell line which is positive for DLX1, we use CRISPR-Cas based knockout, and in these clone again we perform these cell based functional assays. We found decrease in oncogenic potential when we knock down DLX1, which indicate that it has some role to play in oncogenic potential in prostate cancer. Moving forward, then we asked like what are the oncogenic pathways which are involved in this kind of tumorigenesis, which is driven by DLX1. We perform gene expression profiling using the cell line that we created for DLX1 knockout, and we look at some of the pathways, and what we found that when we knock out DLX1, some of the pathways which were down regulated were like proliferation, migration, angiogenesis, Wnt signaling and so on and so forth. So just to validate that, what we perform like we perform immunoblot assays for some of the EMT marker, that is epithelial to mesenchymal transition, and in the knockout clones we see increase in the E cadherin, which is more of an epithelial mark, and decrease in the vimentic, which is more of a mesenchymal mark. Not only that, like when we also perform like a fax based experiment to prove this, that when we knock out DLX1, what is happening to these cells, and we found that more of these cells, they show decrease in the apoptosis, increase in the apoptosis when we knock out DLX1, not only in T2RE1 cell which is a DLX1 positive, the other line which is DLX1 positive as well as R positive also show similar trends. So more apoptosis when we knock out DLX1. Moving forward, since EMT and stemness, they go hand in hand, so the next obvious question was how DLX1 knockout will affect stemness. And when we perform this, sorry, it's like moving too fast. So in this assay, again we use the DLX1 knockout clones, and when we look for some of the genes which are involved in stemness, such as OCT4 and CDK117, that is c-KIT and ATP binding acid G2, we see decrease in the expression. By FACS experiment, we see when we overexpress DLX1, we can see increase in the cell surface expression of CD44, whereas in 2,2-RE1 and VCAP cells with knockout, we see decrease in the expression of CD44. Not only that, another stemness marker is aldehyde dehydrogenase 1A1, and we look for the expression of this particular marker. In RWP cells, when we overexpress DLX1, we see increase in the expression of LDH1A1, and with knockout, we see decrease. So, which indicates that there is a connection between DLX1 and stemness. So now, to prove that DLX1 has a role to play in tumorigenesis, what we did, we implanted this DLX1 knockout clones that we generated in immunodeficient mice and monitored tumor growth. So you can see here, like with the knockout clones implanted in the mice were growing much slower than the scramble control, which has much higher DLX1. And at the end of the study, the mice were like euthanized, and bone and the lungs were isolated from these mice, and the idea was to check the micrometastasis in these organs by performing ALU-PCR, which is specific to primates in the human, because the mice usually they have B1 and B2 repeats. So if we are getting ALU sequences, that means the cells are metastasizing, and you can appreciate here with the knockout you can see very small number of cells metastasized to bone marrow and lungs, which indicate if you ablate DLX1, there is a decreased metastatic potential in these cells, which as a result of that you see decrease in the metastatic cells in the form of micrometastasis reaching to bone marrow and lung. Now, since I told you like one of the most preferred site for metastasis is bone, so we performed experimental bone metastasis where we implanted these scramble control and the DLX1 knockout cells in the tibia of immunodeficient mice and let the tumor grow for 4 weeks. And at the end of the 4 weeks, these bones were isolated, tibia was isolated and subjected to micro-CT. And this is a micro-CT images, and you can appreciate here with this morphometric analysis that the DLX1 knockout clones, they show more bone volume and more bone surface area as well as trabecular number, which is evident here as compared to the scramble control where you have more DLX1. So, which again indicate that higher DLX1 expression in prostate cancer results in bone loss as well as destruction of the bone architecture once the prostate cancer cell they metastasize to bone. Now moving to the mechanistic regulation of DLX1. So I told you like TMPRSS2 is present in 50% of the cases. Then I also told you like 60% of the cases they have TMPRSS2 genetic rearrangement as well as DLX1 overexpression. So we thought, could it be possible that ERK, which is a transcription factor, is driving DLX1, and because there's a big overlap, and with this notion we decided to do another set of experiment where we took this immortalized prostate epithelial cell line, overexpress ERK. So you can see here with the overexpression of ERK, we also see increase in the DLX1 expression. Not only that, we perform some cell-based oncogenic assays, we see increase in oncogenic potential of this cell line. Now to prove that indeed that this ERK is binding on the DLX1 promoter, we scanned the promoter region of DLX1, we found 2 ERK binding motifs upstream of the DLX1, and when we perform chip PCR, we see a significant recruitment of ERK on the DLX1 promoter region. Not only that, we were when we perform a chip PCR for H3K9 acetylation mark, which is a chromatin activation mark, we see increase in the enrichment of this mark on the DLX1 promoter region on the both ERK binding site, and RNA pol was also present, which again indicate that ERK is driving the DLX1 expression in TMPRSS2 ERK positive cases. Now the question is like that is like about the ERK positive cases, and I showed you like 50% of the DLX1 positive cases they don't show TMPRSS2 ERK fusion. Then the question is how in those cases DLX1 is driven. So here we perform another set of experiment. We thought, could it be androgen signaling which is playing a critical role, and we stimulated these cells with synthetic androgen, and as a result of this stimulation we see increase in PSA, which is an androgen target, as well as ERK, and DLX1 was also seen to be increased. Not only that, when we went back to the promoter region and we look for the ARE site, we do see in the first exon of this gene, in the middle of the in the second exon we see there is a ARE, as well as in the intronic region we see AREs. And again we perform chip PCR for ARE1 and ARE2, which are 2 AR response element on the DLX1 promoter region and the introns. And when we stimulated these cells with synthetic androgen, we see significant recruitment of androgen receptor. Not only that, when we treated these cells with enzalutamide, which is anti-androgen, we see decrease in the recruitment of AR. Likewise, we also look for FOXC7, which is a pioneer transcription factor. So usually AR and FOXC7, they work together. It's a pioneer transcription factor. So one is get recruited, then recruit the AR. So we thought, could it be that FOXC7 is also getting recruited? And yes, indeed, when we perform this experiment with androgen stimulation, we do see recruitment of FOXC7 in these cells, VCAF cells. So again, it indicates in the TMPRSS2 fusion negative cases, DLX1 is driven by androgen signaling. Then again, like since androgen receptor and ERG, they both co-regulate multiple genes. So the question was, is this happening in this case also, in case of DLX1? So what we did, we performed a sequential chip, and what we found that ERG and AR, when we pull down this immune complex with ERG and followed by AR, we do see significant recruitment of this complex on the DLX1 ERG binding motif. And using this publicly available ChiaPET data for RNA poll, we integrated the data with the chip PCR data for ERG and AR and FOXC7, which are the regulator for DLX1 and as well as STK estimation mark. We do see a looping, so which indicate that enhancer bound androgen receptor and FOXC7, it's kind of looping with the ERG, which is sitting on the promoter region of DLX1, that is EBM. So with this, like since we understood the mechanism, that the first thing was like to use bromodomain inhibitor. Why? Because bromodomain inhibitor have been used in the clinic for like multiple clinical trials, either as a single agent as well as in combination with anti-androgen drugs. And the idea is because BRD2, 2 and 3 and 4, they go and they bind to the acetylester lysine of the histone, which again recruits other co-activators such as AR or ERG in this case. So it was an indirect approach. So if we use bromodomain inhibitor, we could disrupt this complex, and as a result of that, you can see decrease in the DLX1 expression. So this approach we have used. What we did here, like we treated the cells with JQ1, which is not a very good JQ BET inhibitor, but it's available and it's not that expensive. So for the proof of principle is good. You can see with the treatment of JQ1, we see decrease in PSA and ERG, which was expected. Not only that, because these two regulators are down regulated, that is AR and the ERG, we see decrease in the DLX1 expression as well, which was also confirmed by chip PCR. With JQ stimulated cells, treated cells, we see decrease in the recruitment of ERG as well as acetylation mark, which is activation mark, confirmed by chip PCR. Now finally, performing all these assays. So since JQ was working there, and then we have to confirm that in cell proliferation assays and other cell-based assays, is JQ working or not. What we found that when the cell line is negative for ERG, we see no additive effect with JQ and ENZA, which is anti-androgen. So it's almost similar. Whereas when the cell line is positive for TMPRSS2 ERG fusion, and we treated the cells with ENZA and JQ, there is an additive effect as compared to the JQ alone. Similar effect was observed in this foci assay as well as migration assay. Finally, to take this finding to the mice model, preclinical mice model, we implanted these 2,2-RE1 cells in immunodeficient mice, randomized mice into 4 groups, and treated these mice either with vehicle or ENZA and JQ1 or the combination. Again, similar to the cell-based proliferation assay data, here also we didn't see additive effect, but indeed there was a very impressive, remarkable reduction around, there was around 65-70% reduction in tumor burden. And at the end of the study, we again collected bone marrow and lungs to look for the metastatic cells in these organs, and there was a significant reduction in the bone metastasis and lung metastasis in the group which was treated with JQ alone or a combination of ENZA and JQ. So with this, again, the same thing was proven by doing the immunohistochemistry on the tumor collected from the mice experiment. And you can see here, especially the DLX1 staining, which is where we are more interested, because this is one of the target for ERG and AR. And you can see here with JQ treatment, there's a significant reduction in the DLX1 expression, and which is similar to ENZA plus JQ. So again, this establishes the therapeutic utility of BET inhibitor and anti-androgen, which could be used for impeding the DLX1 mediated tumorigenesis. So this is the conclusion. So we propose 2 models of DLX1 regulation. One is in ERG dependent manner, the other one is ERG independent manner. ERG dependent manner, you have ERG sitting at the promoter region, and this AR and FOXC7 complex is making a loop. Whereas in an independent manner, you have FOXA1 and AR, which is driving the expression of DLX1. And once DLX1 is overexpressed, which is a homeobox transcription factor, it has its own target, which data I have not shown. So DLX1 will go and bind to its target, that is LDH1A1 and HNF1A1. So these two targets, and there are multiple other targets